Hi, it's Professor Bittner, and I am going to give a little intro to how to use this tool. This tool is a simulation, a model. It has built in, it has all of the math, all the calculations that go with population genetics, and these are very complicated calculations. I'm not talking about like a simple Hardy-Weinberg. I'm talking about some very complex calculations, but the good news is you don't have to do any of them. This model has programmed into it all the calculations for how different factors affect allele frequency in a population. And this model actually you can run two populations side by side if you want to do kind of a compare contrast. And it's pretty powerful, actually. You can, we are using a model that has it's focusing on one locus, and you can see over here the frequency of big A, and then the alternate would be the frequency of little a. So the allele frequencies, which you know from your Hardy-Weinberg studies. And so that's on the y-axis. And the frequency of the big A allele can range from 0 in the population to 1. And that means the frequency of little a would go from 1 here at the bottom all the way up to 0 at the top. But this graph shows the frequency of big A. And on the x-axis is time, the number of generations. And there is, like I said, underlying this is a lot of math plus some random number generators, which all play into these simulations. All right, so you're going to have some assignments using this tool. This is not going to be the kind of assignment where I tell you exactly what to click and then you just write something down. You're going to interact with this tool. You're going to play with it. You're going to click on things and see what happens, and then do it again, and then change one variable and click again and see what happens. And you're going to learn by interacting with this tool how population size affects how the allele frequency changes in a population over time. And remember, change in genetic structure over time is the evolution. So you're going to learn how population size is a factor or a variable in evolution. You're going to play with migration. Migration is a factor, a driver of evolution. And you're going to play with fitness. And down here you see some settings for fitness. Fitness is measured on a scale of 0 to 1, where the genotype that has the most chance of survival and reproduction is set to a fitness of 1. And then any decimal less than 1, 0.9, 0.8, etc., indicates that some of these other genotypes have a slightly less chance of passing their genes on to the next generation. Okay? And like I said, you can run two populations against each other, and so you can run two at a time, any way you like. The migration, immigration, these, when you set immigration to 0, then both populations are completely separate. When you add some of the immigration, then they start to intermingle. So that gets kind of interesting. Now, for the purposes of this activity, I'm going to suggest that you keep the generations, somewhere, don't go too small. 400 is kind of the default. I would leave it there. I've noticed when I set it to 1,000, it takes longer for all the calculations to happen. It slows everything down quite a bit. You're welcome to do that, but 400 is probably fine and leave it there. And the other thing we're going to leave alone is the initial gene frequencies. Let's just leave those alone. But you are going to change and mess with population size, and you're going to mess with migration rates, and you're going to mess with fitness, which has to do with natural selection, what's favored, what's not favored. And every time you change those variables, you hit the plot button and it runs simulations based on that. So for example, this is population 1 in the red, population 2 in the blue. If you are colorblind, let me know. There's a way we can make this work for you, but the rest of us are going to see the difference in the color blue and red for generation or population 1 and population 2. And what you'll notice is that if you hit the plot multiple times, even if you haven't changed any of the variables, different things will happen. The same thing won't happen every time. So you really have to hit the button many times to start to get a sense of whether or not there's a pattern in what's happening. So you can see. Now, some things that are kind of important in evolution is everything is going to start, we agreed that we would start with the frequency of big A allele at 0.5 and the frequency of little a at 0.5. So both lines start here at 0.5. But then the frequencies jump up and down and are somewhat influenced by the variables. And what's interesting is when you have something like this where one line hits either 0, if the frequency of big A reaches 0, then that means everyone in that population from that point on would be homozygous for little a. Or if the line hits the top, if frequency of big A hits 1, then everybody is homozygous for big A. So when the frequency hits 1, that's called fixation of the allele. And if the frequency hits 0, that's loss of the allele. But since there's 2 alleles, if we have loss of the big A allele, that means that we have fixation of the little a. And if we have fixation of the big A allele, that would mean we have loss of the little a. And that loss of genetic variability is important in a population, and typically has well, it doesn't necessarily have a negative effect, actually. In some cases it can be adaptive. But you want to kind of notice that as something that's kind of important. So when you keep hitting the button, you'll notice different things happening. And I'll give you some more instructions than this, but there's, you don't need to worry about, oh, did I type the right thing? Did I hit just keep hitting it. Play around with these numbers. I'll give you some more instruction, but you'll play around with these numbers. The things that I want you to leave alone are the initial gene frequencies. Leave those at 0.5. You won't be changing those. And whatever number of generations you pick, I think 400 is good, leave it. And you'll be messing with the other variables, the population size, because that has an effect on drift, the migration, and the fitness, because that's involved with natural selection. So before you even start to look at the assignment that uses this tool, I think you can just play around with it and just hit plot and see what you get and just get familiarized with it.